Good morning, good afternoon, and good evening, everyone, from wherever you're joining us, and welcome to today's webinar titled Getting to the Heart of Cardiovascular Research From Yesterday to Today and Looking Towards Tomorrow, featuring Dr. Melanie White, Heart Foundation Future Leader Fellow in the School of Medicine at the University of Sydney. Today, she will present the methods for assessing cardiac function in the context of pathology, spanning from in vitro to in vivo techniques within her research on cardiovascular disease pathogenesis. I want to take this moment to thank our partners at the American Physiological Society and the sponsor for this event, 80 Instruments, for making it all possible. 80 Instruments creates industry-leading software and hardware tools for life science researchers and educators. For over 30 years, 80 Instruments' mission has been to support scientific discovery, and they are proud to be in strategic partnership with the American Physiological Society to support innovation in the life sciences. 80 Instruments offers a range of solutions for accurate physiological measurements, and with the ability to integrate multiple data sources, systems can evolve as your experiments do, ensuring quality results wherever your research takes you. Okay, and now with that, I am pleased to welcome our speaker, Melanie, to the floor here. Take it away.
Thanks, Sydney, and thank you everyone for joining me today from wherever you're coming from. I also want to take this opportunity to say a big thank you to the sponsors for this event. And I hope we're going to get some really interesting things, some interesting thoughts and discussions going around cardiovascular disease research, looking from yesterday to today and towards tomorrow. So just to give you a bit of a guide as to what to expect from me today, I want to start by having a look at some of the approaches that you probably already use to study cardiovascular diseases, some downstream considerations that you probably are already doing, but maybe some you haven't thought of. And then I would like to talk to you about why ex vivo works for me. I'm going to take you through two of the studies that we've published recently. And then point 4 is some of the challenges that I didn't know I would expect to have to address, but found out the hard way along the way. And lastly, how we've seen Langendorf perfusion change even just over the last 20 years, despite being a really old approach. We're talking about 100 odd years that Langendorf ex vivo perfusion has been used in the laboratory for various applications. We've seen it really changing in the last 20 years. So without further ado, let's have a look at some of the currently available cardiac systems in which we do our main research. So obviously there's ex vivo perfusion applications. These are our retrograde and our working heart approaches. We could also use in vivo approaches, which are things like our coronary ligation, rapid pacing, and aortic banding. Lastly, there are those in vitro approaches that some of you might be using out there. And these are really using the iPSC-derived cardiac myocytes and primary cell cultures. So let's take a little bit of a look at each of them in a little bit more detail. So if we're using a retrograde perfusion, what would we be doing that for? Some of the traditional applications for retrograde perfusion, Langendorf, is for global ischemia reperfusion injury, but also pharmacological testing. And this is because one of the advantages of retrograde perfusion is the precise timing with which we can either apply ischemia, reperfusion, or even that pharmacological intervention into the heart. It's also devoid of neurohumoral effects. Some of the limitations, however, that should be considered is that it's a crystalloid perfusate, and this can lead to increased osmotic pressure within the cardiac myocytes themselves. Alternatively, in the working heart, as you can see, the applications are very similar, but the advantage here is that we're able to manipulate preload and afterload really nicely, and we're actually using physiological flow. The limitations with working heart is it's incredibly expert. You need an expert to really make sure that you're getting the most from your working heart system. Both of these systems allow us to assess left ventricular functionality without those neurohumoral effects, which can be important depending upon your biological question. Now let's head into in vivo. So obviously in vivo is a much closer environment to the clinical setting, especially if you're working in a preclinical environment, which is where I come from. So if we're doing a coronary ligation, the application is something like myocardial ischemia or infarction. The advantage, obviously, is we're looking at the longer-term outcomes and the systematic effects of the effect of the heart. The limitations, obviously, a high degree of expertise. There can be variability without such expertise. And ultimately, we're looking for our animals to survive. So if we don't have survival of these animals, our experiments are not meeting the criteria we are setting. Secondly, rapid pacing. This is obviously something that is used really nicely for tachycardia and heart failure. The advantages are obviously its clinical relevance, but also it's rather simple to set up. The limitations, it can be a reversible form of tachycardia if that stimulus is not continued. Lastly, aortic banding. This is where you might be using it for applications including aortic stenosis to develop hypertrophy. The advantages, once again, it's clinically relevant and it provides systematic effects. The limitations, once again, because this is a cardiac surgery that is required, you will need, there's a high degree of expertise that is needed to ensure success, and there can be variability depending on where you put your suture from experiment to experiment. But obviously the real feature that sits across all these in vivo approaches is that it gives us the capacity to assess functionality in a much, much longer term. So we can see how our phenotype is adapting to our cardiac event. And it allows us to look at the extent of remodeling and provide that nice clinical relevance. And lastly, if we look at some of the in vitro models, obviously there's been a lot of development recently in the iPSC world. It gives us a great application for things like pharmacological testing. The advantages, we can actually use human samples here, and as well as other numerous preclinical models as well. Some of the limitations, obviously, it can be tricky to achieve cellular maturity in these cells. Expertise is obviously another important part of that, but cellular diversity can also come into a potential limitation, as well as how the cells actually organize themselves by comparison to what we would expect in a non-in vitro setting. Lastly, our primary cells, some of the applications that they've been used for, and this is obviously an older technique, is global ischemia, reperfusion, and also pharmacological testing, as well as a lot of electrophysiological work. Some of the advantages include the vast array of those downstream applications that primary cells have been used to assess or to be assessed using. Limitations, expertise. I've certainly had my fair share of failed primary cardiac myocytes, also the recovery, but also, and finally, the longevity. But the overall advantage of our in vitro system is that we can use both clinical and preclinical cells to take an in vitro approach. So we need to consider, with these different samples and these different approaches, where we're deriving our biological tissue from. So when we're doing, and what sort of considerations we need to make. So if we are performing an ex vivo experiment, then we're looking for short-term outcomes. This is really what is happening at the there and then. We're not getting that we're not getting a long-term functional recovery here. And it does require a terminal procedure to generate this tissue. With in vivo, a consideration is not only those approaches that I spoke about, but you can also use diet or gavage treatments or water to deliver therapeutic options into your in vivo model, which can be really interesting. But the thing we're really looking for here is animal survival. And that's a really important consideration when moving towards in vivo models. Lastly, with in vitro systems, especially with those primary cardiac myocytes, here we're going to need to be able to treat them with collagenase. That has a consideration to keep to be at the front of our mind, that to what extent is that collagenase actually impacting the cardiac myocyte from its moving it from its physiological function. It requires a terminal procedure or alternatively explant tissue. Okay, so the way I see my science evolving over time is that my biological question will ultimately sit in one of these three silos. And it's not to say these silos don't talk to each other, but it makes it helps me define where my research sits. So my research is either going to sit into a discovery platform, in a discovery silo. Here, the sorts of questions I might be asking in a discovery component, what are the key processes, be them molecular or cellular processes, that are defining the pathogenic process? Because I'm interested in what is actually defining that biological process or those pathogenic changes, I really want that to be a biologically uncomplicated system. If I move into a validation biological question, here a kind of question I might be asking is, do these changes actually occur in a variety of settings? Here, it gives me the capacity to go back to my discovery with my targeted list of things that I'm really, really interested in, and now add in the complexity that population studies bring to it, and introduce that level of biological complexity to see if my discovery targets are still relevant. Lastly, in my translation silo, here I'm asking questions about whether these discovery targets that I've been looking for and then gone on to validate with biological complexity actually translate to a clinical setting. Obviously, there's a lot of diversity in our clinical cohorts, in our clinical patients who might be presenting with cardiovascular disease. And I'll talk to one of those examples in an example of my own. But here, we really want our translation to be able to design a therapy that's going to alter that pathogenic driver. So here, we're really kind of taking it all the way. And you can see how even though I've kind of siloed them, they do follow from one to the next to the third. Okay, so obviously going beyond where we're going, what sort of tissue or what sort of approach we're going to be using to ask our biological question, we obviously need to test our biological question. And we're going to test it either using multiple measurements, be them functional measurements, where we might do left ventricular developed pressure, for example, using Langendorf perfusion. We might do echocardiography to look for cardiac contractility, pressure volume loops, MRI, electrophysiological, or even fluxomics. We would obviously want to complement that with some sort of structural measurement. Histology and immunohistochemistry are really nice ways to go about that. But also there's things like living myocardial slices and cardiac organoids to give us that structural component that we will want to measure in our biological context. Lastly, biochemical measurements. And this is where we might be considering Western immunoblots, ELISAs, any of the omics approaches, be it genomics, transcriptomics, proteomics, flow cytometry, or even assessing biomarker release. So obviously I've mentioned that we want to be taking multiple measurements across these dimensions. The measurements that we are using to really show function, structure, and biochemical adaptations in my laboratory are to use our Langendorf perfusion and echocardiography to see how the heart is functionally adapting to our pathology or our change in biology. Then we're going to do structural measurements by using histological approaches, But our real workhorse, and this really reflects the fact that we are a molecular biochemistry laboratory, our workhorse is really our mass spectrometry platform, which we complement with Western immunoblots. Okay, and the thing that we like to consider is that anytime we can increase the dimensionality of our measurements, that's taking multiple measurements in a considered and parallel approach, we allow our biology to drive our research and to define what is important rather than us coming in with a preconceived notion, and it allows for that unbiased discovery component of our cardiovascular research. Okay, so as I just mentioned, the vast majority of the work that I do is definitely in that discovery silo. So here we're really looking to make sure that we are understanding our biological changes or pathogenic changes, and that we can link to our molecular phenotype. And to do this, we look at these components that take us from our genome towards what we see the heart actually doing, which is obviously our phenotype. And for this, we really look to interrogate the proteome, the lipidome, the metabolome, and what I call the modificome, which is a horrible word, and I do apologize. But this is our post-translational modifications of proteins. And it's all of these dimensions where we can get changes in protein structure, function, and outcomes that really help us define the phenotype. This is especially important in the heart because we're not expecting vast amounts of protein turnover. So you probably have heard that a lot of these omics approaches are regarded as what we might call a fishing trip. But instead, I like to think of these approaches instead of as being a fishing trip, but actually allowing our biology to define what is important. So it's not just going out on that big fishing trip, but it's actually defining what is the catch of the day. But as you can imagine, to ensure that our phenotype is adequately represented in the measurements we're making using these approaches, we really need to ensure that the only thing that is defining our biochemical signature is our biology itself. So we need to ensure we have good sample reproducibility to ensure that what we are looking for and what we're going to follow up in our validation and our applications approaches are reflective of the biology, not just our poor sample handling. So we need to have a really stringent approach to how we develop these samples. So just to give you an idea of the workflow that we follow, we start with our sample of interest. Obviously, in my particular case, this is cardiovascular tissue. We extract proteins, turn them into peptides that we can either take a labeled approach or a label-free approach. We then assess our analytes, be it peptides or lipids or metabolites, by liquid chromatography, tandem mass spectrometry. We perform some data analysis to allow our to allow so many measurements across so many time points to really allow the biology to speak for itself, rather than coming in with a preconceived notion as to what our answer will be. And that allows us to generate an appropriate hypothesis for further testing, which is where we enter our validation silo. So, as I mentioned, the requirement considering this workflow is that I need to be able to make precise measurements that really only reflects that biological question that I'm interested in without any confounding biological interference. And as a result, I need a really, really tightly defined model, and I need a really, really tightly defined way of generating my tissue. And the solution for me has been ex vivo Langendorf perfusion. Why is this? It's because of the affordances that ex vivo Langendorf perfusion really provides. So it provides me with instantaneous functional measurements. My omics approaches are what I would call a snapshot technique. So I'm getting an understanding of the biology inside the cell at the moment at which I stop everything, which I do with a drop into liquid nitrogen. So therefore, I need to be able to measure my function at that exact same moment. And there's literally maybe 2 seconds between having a balloon inside my left ventricle and that heart being submerged in liquid nitrogen for further biochemical assessment. The next affordance is obviously the capacity to have constant flow or constant pressure delivered to the heart. It also allows me precise induction of when I give the heart a therapy or when I give it hypoxia or ischemia. And this is really important because I need to make sure my samples are really reproducible. And if I can't do that, then I'm introducing an extra variable that my techniques might very well pick up. And that's one of the things I have learned along the way. The next thing, the next affordance that my Langendorf allows me to, or provides, is it gives me the capacity to collect perfusate, to assess biomarkers that are released from the heart that might validate my method, my Langendorf approach. Things like the release of troponin I would be a good one. This is a perfusate option. So the hearts are not being perfused with blood. And for me, that's an important consideration because the blood on its own has its own separate proteome, lipidome, and metabolome. And I only want to be looking at my heart's phenotype. So I want to be able to remove those confounding factors. And lastly, speaking of confounding factors, I'm also removing every other organ and neuro and humoral effect, which means that the only biology I'm seeing is coming directly from the heart. But as you can imagine, not everything is always on the positive. There are some limitations that I need to come into my experimental decision tree with. And I need to be comfortable with these as being limitations before proceeding. Because if my limitations outweigh my affordances, it might not be the optimal approach. So the limitations that I am well and truly aware of with my Langendorf approaches to ex vivo perfusion, obviously it's a preclinical model. Am I willing to take this as a limitation? I am, because I'm in my discovery phase. The second limitation is that it's a supraphysiological glucose solution that is being used to energize the heart. Am I okay with this? That's a yes and no, depending on my biological setting. And the last one that is a limitation is obviously the progressive but slow decline in contractile and chronotropic function over time. Am I okay with this? I'm okay so long as I make adjustments that are appropriate so that it doesn't impact my outcomes for my following experiments. Or if I do decide to take that, then I need to have appropriate controls. So with all of that in mind, I'm pretty happy to proceed with ex vivo Langendorf perfusion in my setting. As I mentioned, I love my data. I can't get it. Well, if I haven't mentioned it, I love my data. And the more measurements that I can make that allow me to better define the biological signature I'm getting from my omics approaches, the happier I am. The Langendorf, as you can imagine, allows for so many data points to be measured. And that's one of the key reasons I keep coming back to Langendorf ex vivo perfusion. I can measure things like my perfusion pressure, which is also an indication of aortic pressure. I can measure the flow rate. I can measure the heart rate. I can measure the diastolic and systolic pressure, and then I can calculate my left ventricular developed pressure from that. I can take EKG readings. I can use oxygen sensors. There's just so many measurements that I can take that I can then use to assess any potential outliers that might come through that I wasn't expecting. And trust me, they do happen. Okay, so I've come in with my biological question. I've decided that it's in my discovery silo. I'm going to do some Langendorf ex vivo perfusion. But what are the last couple of decisions that I need to make before I put a heart on my perfusion rig? The first one is, what's the species to be used? There are our rodent models, be it mouse, rats, guinea pigs, or larger mammals. We've used rabbits on these systems. Are sex differences going to have a role to play? Am I interested in looking at male versus female responses? If I'm doing normoxic function, there's probably, a rationale to have both males and females in my cohort. But if I'm looking at something like infarct studies, then I need to be aware of the fact that females will have smaller infarct zones. What about age? Age can obviously have a difference. And this is something that we're going to see with our left ventricular developed pressure. And that's going to happen both in normoxic or continuous perfusion modes and also post-ischemia. And age differences can also have an impact on flow rates. So we always want to make sure we have our studies age matched. What about our choice of anesthesia? This is another lesson I learned the hard way, and we'll come back to it in a little bit. But it's important to know that your options that are available are either inhalants or injections, but pick one. By all means, only pick one. Don't try to go across multiple. You also want to consider what your downstream applications are. As I mentioned, my downstream applications are my snapshot approaches. So this is my omics approaches that take me as close as I can to the phenotype. Lastly, are there any molecular adaptations that I need to be aware of that might be detrimental in an ex vivo Langendorf perfusion, where I'm perfusing my hearts with a supraphysiological glucose concentration? Spoilers there. All right, so I'm just going to play a video here for you. This is our Langendorf in action. There we go. It still astounds me, and I've been doing this for a very long time. But the kind of quick, brief run sheet of how we do our Langendorf experiments. We start with a pentobarbital injection that's given intraperitoneally. We are looking for a loss of pain reflex before we start our surgical approach. And the first thing we identify is we open up the abdominal cavity and we identify our renal artery. And we deliver heparin directly into that renal artery, just so that we don't get or we minimize our risk of blood clots that could impact the cardiac function as we progress to our ex vivo perfusion. Once we've opened up the diaphragm and we're in the thoracic cavity, the heart is excised as quickly as we can before it is plunged into ice cold saline solution. And the amount of time that we take from that point until the heart is being perfused, or it's attached to the cannula and being resuscitated with our Langendorf ex vivo perfusion, is no more than 2 minutes. This is something that is really key for us, because otherwise we're starting to look at the effects of ischemia on our heart from our omics approaches. Okay, and that's one of those biological confounding factors. So this is one of the reasons we use Langendorf. Okay, so we make sure we have flow already established when we're connecting the aorta to the cannula, but we gradually increase it to our maximum depending upon the size and the age of the hearts. And with our rat hearts, we normally take it up to 15 mils per minute as a starting point. We go for a constant pressure, and we give it a 20-minute baseline initiating component, and I'll discuss that in just a second. The last thing we do is we insert our balloon into the left ventricle, and that is set with an afterload of between 10 to 15 millimeters of mercury. Okay, so why are we doing a baseline period? This is something that we've been using now for quite a long time, and it's the period initially after we've ligated the heart on the cannula and we've commenced that increased flow rate. Depending upon how long the heart is going to need to be Langendorf perfused, that baseline period will run for either 15 or 20 minutes. And the reason we do it is it allows for the washout of those metabolites that are released during that short period of ischemia. Like I said, we try to make sure the amount of time the heart is not being perfused in the animal to when it's being perfused on the Langendorf, and we try to minimize that amount of time to no more than 2 minutes. Okay, so from that small 2-minute insult, we want to wash out any potential metabolites that have been released from at-risk myocytes that might be releasing things like lactate. And we're going to wash that away and ensure the heart is as healthy as it can be. Okay, it also gives us the capacity to start to look at the functionality of each individual heart, and allows us to consider what some exclusion criteria might be. Why would we need to have exclusion criteria? Well, it just takes a zoom into our data to actually see what might be happening. Some of the reasons we might need to exclude a heart from a study, it might be that the amount of time, that short duration of ischemia has actually been a little longer than our 2 minutes. Obviously, this has the capacity to impact how the heart is behaving because it's had an ischemic insult. If we have an end baseline rate pressure product of less than 26,000, we find that this is a good indication that, or if it's fallen below 26,000, there's something either gone wrong with the heart or with the with the setup of the instrument, and this just happens. It's normally because of the heart that this happens, but it's normally a good indication that the heart is going to prove to be an outlier using our really sensitive techniques downstream. Another exclusion criteria is if our flow rate at that preset of 15 gives us too high of a pressure, aortic pressure on an individual heart. Obviously, because it's constant pressure, that would mean that the flow rate will reduce. So if we have a flow rate that's less than 10 mils per minute, we ultimately pop that into our exclusion criteria. And the last one are our arrhythmias. And you can see a really nice example here. Unfortunately, this heart was an outlier and it never made it to further biochemical study. Okay, so I want to take you through very quickly two of the projects that we've had published in the last little bit that really reflect why Langendorf is a key workhorse in our laboratory. Okay, so there's a lot of work in the scientific field about reperfusion injury. And obviously, reperfusion injury occurs after ischemic insults, and it has the capacity to reduce contractile recovery. So we went about looking at antioxidants and their role in restoring contractile function and contractile recovery. So a few of the things that we've noticed over the past 20-odd years is that despite having that depressed contractile function following ischemia reperfusion injury, there were very few changes to the proteome. What does that mean? It really means that as we expect with the heart, there's not a lot of protein translation, protein expression that is really giving us our physiological output or our change or our pathological change. So to us, this indicated that our pathology was being driven by something else. And the thing that we originally turned to in this particular study was the effect of protein post-translational modifications, keeping in mind that these could be regarded as functional decorations that allow a protein to adjust in a really quick way to impact structure, function, binding partners, and its overall capacity to do its normal function. So we used our Langendorf system really nicely to infuse our antioxidant known as mercaptopropanol glycine, or MPG. And this was added solely in our reperfusion protocol to improve contractile function and to assess our contractile function. So you can see here our different protocols that were considered in this paper published by one of my really talented PhD students, Alex. And each one of our hearts had a 20-minute baseline period. We have a group that went for 50 minutes of non-ischemic time control, and this was our physiological control. We had a group that was exposed to ischemia, 15 minutes of ischemia. We had a second group of hearts that was exposed to 15 minutes of ischemia, followed by 15 minutes of reperfusion. And lastly, our MPG treatment was delivered just in our reperfusion period. So what you can see here, and this is post baseline, you can see that our Langendorf really allows us to clearly define how our function is being impacted by what we're doing to the hearts. So you can see here our non-ischemic time control hearts are very happily pumping, as indicated by our rate pressure product, which considers our left ventricular developed pressure and our heart rate. But as soon as we send our hearts into ischemia, we get this this massive decrease in the contractility capacity of our hearts within the first 5 minutes. That obviously remains at 0 for the remaining duration of that ischemic insult. But really, really nicely, by including MPG in our perfusate, we're able to restore functionality to hearts that would otherwise show about a drop in about a return to baseline perfusion of return to baseline contractility of 75%. Okay, so now that we've seen this, we obviously want to identify what our targets might be. Considering that MPG is an antioxidant, we want to consider what those targets are that MPG is protecting against. So doing global redox modification system-wide analysis is not something that there's not a lot of commercially available approaches. So we took it upon ourselves to develop some methodologies to do this. And we looked not only at the reversibly modified cysteines that were being reversibly oxidized, but we also looked at their irreversibly oxidized counterparts. So this is where we don't have the capacity to reverse that oxidative process. So this is things like sulfinic and sulfonic adaptations to our cysteine peptides. And what we were able to see was that MPG was able to prevent over-oxidation just by being there during our reperfusion insult, which came after our ischemic insult. And the proteins that we identified that were particularly at risk of being over-oxidized, were the enzymes, and they were really close to being the cysteines that were involved were actually quite interesting. So this was our discovery, and we wanted to obviously validate it and quantify what we were seeing occurring with these cysteine peptides. So we needed to develop a method to validate these observations that we'd made. And Alex did this by obviously appreciating that there's very little out there. There's no real Western blots or antibodies that allow us to assess total cysteine levels of oxidation. So we had to design something here. And this is where Alex really did a fantastic job of considering what of our approaches might fit into that point in our silo to allow us to validate. And we turned back to mass spectrometry, and we developed some parallel reaction monitoring approaches to allow us to quantify how those cysteine residues were changing. But it also allowed us to functionally interrogate it by looking at a parallel approach with metabolomics as well. So this here is the TCA cycle, and you're probably going to struggle to see the actual components of the TCA cycle, but here we have our enzymes and here we have our metabolites. I think that's right. Yeah. And what Alex was able to very nicely do is to assess the proteins and their partner metabolites to show that when we do have changes at specific cysteine sites with ischemia reperfusion injury that was protected by the addition of MPG, the metabolites matched up. And this was what allowed us to not only quantify the changes, but to validate that those changes were also having a functional impact in the heart. And that was a really nice study that Alex performed. Our second study that I want to take you through is one that's a longer question that's coming from my laboratory, and that's looking at the link between type 2 diabetes and cardiovascular disease. And it's obviously important to consider that these are independent risk factors. That type 2 diabetes is an independent risk factor, and it's a modifiable risk factor for cardiovascular disease. But importantly, for diabetics, they're at not only a greater risk of having a heart attack, but also having worse outcomes following a heart attack. Even before a diabetic shows up with elements of a heart attack or signs and clinical features of a heart attack, diabetic patients actually have decreased contractility. And this contractility is independent of those classic cardiovascular signs and symptoms that would otherwise predate an acute myocardial infarction or a heart attack. Things like coronary artery disease, hypertension, and changes in the valve functionality. And this really reflects those early changes in metabolic fuel that is delivered to the heart. So for this longer term project of ours, it has been our goal to understand what are those changes that are occurring in the heart with diabetic cardiomyopathy that really mean that these hearts aren't going to recover after a single cardiac event. So here you actually get to meet Alex, and this is my other PhD student, Desmond. And we went back to our discovery platform, and we really wanted to make sure we were doing an integrated approach. So we used our multi-omics series here to really interrogate the phenotype across 3 dimensions, and that was our proteomics, our lipidomics, and our metabolomics. And for us, it was really important that we had sufficient tissue that meant that these were parallel experiments, and we could interrogate a single specimen across these 3 dimensions that could then be re-evaluated with the data that we collected from our Langendorf as well. So we had a requirement that would allow us to do this. Obviously, we wanted to pair all of these samples. We wanted to be able to match our results from these individual omics assessments. And therefore, we needed to make sure we had sufficient tissue to start with. And as a result, we moved into a rat model. So just very, very quickly, in a preclinical setting, obviously we have to generate a phenotype. And we can do this by numerous different ways. For diabetes, as an example, we can use genetic models of obesity. Things like the Zucker diabetic fatty rat are really kind of classic approaches to developing that overarching phenotype of diabetes. We can use diets to start to introduce our phenotype, be it a high fat or complemented with sucrose. We can also generate a diabetic phenotype by introducing pancreatic insufficiency through the injection of a pancreatic toxin like streptozotocin. There's some really nice new genetic engineering approaches. And lastly, our combined approach. But once again, it all has to fit into our discovery, validation, and our translation profile. So considering this, we need to be able to have a phenotype, our overarching phenotype of diabetes, that is going to be developed through a similar mechanism as what we're going to see in our translational studies. Why do our diabetics come in to the clinical cath lab? That's because they're, largely speaking, obese and they have pancreatic insufficiency. So I want to make sure my discovery pipeline reflects that clinical phenotype rather than some sort of genetic manipulation that although it gives me the diabetic phenotype, doesn't actually reflect what my clinical cohort is going to present with. And that's why I actually go with a different approach to those genetic models that a lot of people use. And here I want to reflect the obesity that these patients present with using a high-fat, high-carbohydrate diet. And I want to reflect their pancreatic insufficiency by developing by introducing that pancreatic toxin using STZ. Because otherwise my downstream approaches could be giving me, instead of the biological cues that are coming from these pathogenic drivers, they could be telling me about the off-target effects of what happens when you silence a gene. So what we were able to see taking this approach are lots of different changes that were happening because now we're in a chronic model. As I mentioned, we like to let our data speak for us rather than coming in with preconceived notions. So we took a triangulated approach to really overlay our lipids, proteins, and metabolites and let the biology define what was important. And it led us to understand that in the diabetic system, we were getting increased fatty acid delivered to the cardiac myocytes, and that was having an impact on beta oxidation inside the mitochondria, as well as downstream effects on TCA cycle. Okay, so some of the lessons we've learnt using Langendorf systems. The first big lesson that I learned was in 2002, and that it's not all hearts are created equal. And this here you can see is a really nicely separated two-dimensional gel electrophoresis sample of a non-ischemic time control heart. But when you do a deeper dive, what you see are these tiny little alterations in proteins that were really interesting. And we found that that was a fragmentation of that occurred to myosin light chain 2. But I had this one outlier. No matter what I did, no matter how many times I ran that gel, that heart was always a problem. And it turned out that despite meeting my end rate pressure product expectation of 90% maintenance of function. It didn't meet my initial RPP expectation of 27,000. So that heart was then excluded from the study as it didn't meet that expectation. And it allowed me to introduce exclusion criteria. The second lesson that I have learnt the hard way is that not all anesthetics are created equally, and I learnt this by doing phosphoproteomics. So here we're looking at how the proteome is adapted or is adapting to a phenotypic, adapting to a biological outcome through signal pathways. And what did we learn? Well, what did we see actually? We saw that there was a 20-fold change in pyruvate dehydrogenase phosphorylation just from moving between pentobarbital and isoflurane. So we learnt not to move between anesthetic agents, but just to pick a single one. And our last lesson in 2019 was that the metabolome is a rather dynamic environment. Metabolomics is where we're measuring our small molecules. And what did we find? We found that the changes in our metabolome can actually just sometimes reflect what is happening in our Langendorf system. So if we've got a metabolite building up, it could reflect the fact that we don't have we're in an ischemic insult and they're not being released from the cardiac myocytes. Or it could just reflect the use by the cardiac myocyte, for example, carbon sources and ATP. So I mentioned that I've been doing Langendorf perfusion for quite a while, and we've seen some real changes happening in the last 20 years. The first part is obviously with data recording. Back in 2000, when I first started my PhD, we were using this a piece of instrumentation, which is a grass recorder, and it can also be used as a lie detector. But in our particular case, we were recording cardiac output. It relied on ink and paper, and to do data analysis, that was incredibly time consuming because you would literally flick through paper. Thankfully, in 2008, we moved across to the Power Lab, which allowed us to take analog measurements and convert it to digital data. As you can imagine, this is an automated system, it's modular, and it's a very much a plug and play environment rather than the old grass recorder, which was a bit finicky. The other important component from the Power Lab is we can start really wide and we can see the entire data, we can see the entire perfusion protocol, and we can also zoom in. So we can start to see small changes in cardiac function, which we will definitely see as changes in our signatures by our omics approaches. Okay. The last thing that it allows, that Langendorf allows us to do, is it allows us to either take endpoint analysis as well. We can also do a temporal analysis, and we can see how function changes over time, which can be really important depending on what your biological question is. Okay, the next development that we've seen over the past 20 years is how the system is actually updated. So we originally started with a Rudnotti system. It's an incredibly complex series of parts that all have to communicate and work together well. And we actually ended up needing a few backyard hacks to make sure the system remained functional. But as you can imagine, this is a system that has a deep history involved with it. And one of the things that we needed to do when one of our pumps broke was we relied on a 6-foot change in where the heart was located, as you can see down here, to where our bench was, so that we could ensure that the perfusate was being delivered at the appropriate pressure. So there, we could make it work. But unfortunately, it was packed away in 2007 and it never regained function. In 2008, we moved across to the Pan Lab system, which was fantastic because it was very compact. It sat on my desk very nicely. It was ready to be used straight out of the box. And it allowed us, with the use of the pump, to deliver either constant pressure or constant flow to the heart. However, over time, because it was a Perspex system, it began to craze. We required inline filters to make sure the perfusate was being delivered to the heart in its cleanest fashion. We also noticed that the dead volume between the reservoirs, where the heart was, and the reservoirs where we had our perfusate and the junction block, was really long and unnecessarily long. So last year we moved across to the AD Instruments rodent Langendorf system, and it has really been a game changer. As you can imagine, it's straight out of the box, ready to use, same as the PanLab system. It's compact and modular, same as the PanLab system. But it gives us capacity to do more. And this really reflects the borosilicate glass. And that's been a real game changer for us because it is autoclavable. It also means that we have a clear line of sight into the reservoirs because we're in a glass system instead of being in a Perspex. So my favorite features, I'm going to play this little video. So as you can see, the heart is very happy on this system. It's compact, it's desktop. I can see straight into my system because it's a borosilicate glass system, and my reservoirs are nicely closed. It has a spill tray, which we can see here, and I get to collect lots of data. Just as a comparison for how compact this system is, you can see here that this is my Langendorf system, but over the back end, this is a working heart system. And you can see it's pretty much touching the ceiling. Okay, so my favorite features of the new direct perfusion core that AD Instruments has just released. The 4 sensor points coming directly into the perfusion core. You can see them. You can see all 4 of them just there. It has the capacity to recirculate my buffer back to the reservoirs, which you can see here, which means that my buffer is never being delivered to the heart, even after an ischemic insult, without being adequately gassed. It also means that I'm not wasting any of my perfusate. There's a smaller volume between my reservoirs and the junction block, which is important. I've got a built-in bubble trap, which you can see sitting up the top here, and I've got really nice heat exchange as well in the system. And the thing that I think that is really important to keep in mind is that any of our systems, they need to be robust, flexible, but also modular. And that's what's going to make sure we take our systems from today and can continue to use them as we move towards future downstream applications. So in my lab, where are we going? We are moving to treat diabetic hearts with antioxidants. We're going to be using our new AD Instruments system to infuse hearts with labeled metabolites to do some fluxomics and perfuse our diabetic hearts with different carbon sources. And with that, I'd like to take the opportunity to not only thank the sponsors of this event today, but also my funding partners, including the Heart Foundation, Ramashodi, and the Australian Research Council and NHMRC, and not to mention the integral parts of my laboratory, without whom this work would not be achievable. And thanks, Sydney. I'm happy to take any questions.
Great. What an interesting and detailed presentation, Melanie. Thank you so much for that. We have reached the top of the hour, so hopefully you can stay a few more minutes to get to our questions. Melanie, there have been a lot that have come in, so let's get right into it. In cardiomyocyte or fibrocyte-specific knockout animal studies, what would be the easiest method to isolate the cells from animals, and at what age group animal would you recommend?
It's a great question, Sydney, and I would love to give a more detailed answer, but it just reflects the fact that it's not my main workhorse. So I would say that it's just really important to be consistent. Consistency is key. So age matched is obviously important. Fibroblasts are one of those key cell types that you need to consider the age of the animal when looking for fibroblasts. So I'm I'm sure that you'll make the right choices based on the scientific literature.
Okay, fantastic. Thank you for that honesty and information. Do you have any tips for immobilizing the heart during recordings? Example, monophasic APs.
Yeah, look, the new system that we've got, that we got from AD Instruments, it's got a specially designed cap which actually has these 4 ports beyond the 4 ports into the direct perfusion core, which allows us to securely insert the electrode set that you can also purchase. And because there's those 4 reference points, it also allows us to secure the heart and immobilize it. So I'm really hoping that that is one of the ways that we're going to be able to immobilize the heart really nicely.
Okay, great. Thank you. Our next question, for in vivo AMI study, how do you determine that your IR protocol is successful?
Yeah, look, that's a great question. And it really depends on what your endpoint is. If you're looking for an immediate definition as to whether you have success in your IR protocol, then a really nice way would be to take a small plasma or small blood sample and to test for your biomarkers of ischemia reperfusion injury, like your troponin I. So classic clinical approach. If you're looking for midterm, so you want your animal to be surviving any kind of functional assessment, then I'd be looking at echocardiography as a really nice way to assess whether my ischemia reperfusion protocol has been successful. But if it's an end stage, so where I can perform a terminal procedure, then I would be looking for something like TTC staining as an indication of necrosis.
Excellent. Thank you for the breakdown. That was great. Okay, we've received another question asking, given differences in conduction channels, heart rate, and blood pressure, how well do rodent perfused heart studies translate to human disease models?
Look, it's a great question, and there is no way I would ever go straight from a rodent into a clinical setting without first doing a validation. So a proof of concept. Like I mentioned, I really kind of try to pull out what we do into either a discovery, a validation, or a translation silo, and everything that I do in a preclinical rodent setting in my discovery, which needs to be really biologically clean without confounding factors, needs to be validated in a less pure sample where I start to get some population variation. So there's no, anytime I'm doing a preclinical model, it has to be validated in a more challenging environment.
Okay, great answer. Thank you so much. Let's see here. Someone has sent in that they're just getting started with heart perfusion studies. Could you give some tips, tricks, or advice for beginners?
Yeah, practice. These things definitely improve with practice. We really there are key critical points, so pinch points that I like to point out, and that's really how long it takes to get the heart from perfusion in the body to being perfused using your ex vivo system. We really try to get that down to less than 2 minutes. That takes time. One of the other things is the importance of how well you're making up your buffers. So we literally have gone from one building to the next, changed our water, and our hearts started dying. So if all of your hearts are dying for no good reason, keep an eye on your water, and also make sure you have an inline filter. Any kind of particulates or dust, that becomes a real problem when you're thinking about the vasculature of a small rodent system like a mouse or a rat.
Great. I'm sure that advice will be very helpful. Thank you for sharing. Okay, let's get one last question in. We have gone a bit over time. Hopefully, most of you are still able to stay with us, but keep an eye out for a Q&A report that should be coming out in the coming weeks that Melanie will be able to provide some answers for. So, what are the biggest challenges I should expect to face with these applications?
Wow. Okay, consistency. I think you really need to be consistent. There's nothing worse than not having age-matched samples or clearly defining sex contributions, and the more data points you have, the better you are at being able to find your potential outliers. Like I said, back in 2002, I spent way too much time looking at that one heart just to find out that it was hung by someone who had very little experience hanging a heart. So it was more than 2 minutes.
Okay, fantastic. Thank you so much for all of those answers, for your wonderful presentation. It was such a pleasure to have you here with us, and I hope you enjoyed yourself today.
Thank you so much, Sydney.
In closing, we hope you enjoyed this Inside Scientific webinar produced in partnership with the American Physiological Society and sponsored by AD Instruments, and I hope you enjoy the rest of your day.